Good morning. In this session, I'm going to talk on plant growth regulators. Plant growth regulators. First thing, what is growth? Growth is an irreversible change in mass. An irreversible change in mass. That is, increase in size, volume, and weight of any part of plant's body. It means quantitative increase in plant body, for example by cell division, cell enlargement. See, this is the growth. Growth is or it can be defined as irreversible change in mass. Next, what is development? Development is nothing but an irreversible change in state, an irreversible change in form. It means the qualitative change in plant body. For example, follow my cursor, seed, then it becomes seedling, then vegetative growth, then maturation, then flowering, then reproduction. See, you cannot reverse back. It is a irreversible change in state or form. That is, seed becomes seedling, then vegetative growth, then maturation, then flowering, and so on. See, the plant growth and development are under the control of two sets of internal factors. Number one is nutritional factors such as the supply of carbohydrates, proteins, fats, and other constitute the raw material for the growth and development. The second thing is proper utilization of these raw materials is under the control of certain chemical messengers which can be classified into hormones and vitamins. See, these two are the internal factors which controls the plant growth and development. The term hormone is derived from a Greek root hormou, which means to stimulate. So hormone andre to stimulate ಅನ್ನೋ meaning ಇರೋ Greek word ಇಂದ ಬಂದಿದೆ. Of course, as you all know, majority of the science terms are derived either Greek or Latin. Thiemann in 1948 suggested using the term phytohormone for the plant hormones or hormones of plant. Phytohormones are organic substances produced naturally by the plants which in minute, very low concentration. So phytohormones are nothing but the organic substances which are produced by the plants in a very low concentration. These phytohormones involve in either increase or decrease or modify the growth and development of the plants. So phytohormones andre organic substances plants produce ಮಾಡ್ತಾರೆ in a very low concentration. This involves in the increase or decrease or modify the growth and development of plants. These phytohormones also termed as growth hormones, growth promoting substances, growth substances, growth regulators, growth factors, and so on. See, the plant growth regulators. Plant growth regulators refers to natural or synthetic substances which influence the growth and development of plants. For example, indole acetic acid. It is an auxin. It is available in natural as well as synthetic forms. Indole butyric acid, that is also an auxin, always synthetic. All plant hormones are growth regulators. So all plant hormones are considered or involved in the growth of the plant. They are nothing but the plant growth regulators, but all plant growth regulators are not plant hormones. All plant growth regulators are not plant hormones. Next is classification of plant growth hormones. First classification is based on the origin of the hormones. Origin means whether it is natural or synthetic and so on. First one is natural hormones produced by some tissues in plants. also called as endogenous hormones, for example, indole acetic acid. Second class is based on the origin, it's a synthetic hormone produced artificially and similar to natural hormone in physiological activity, also called as exogenous hormones. See the difference between endogenous and exogenous hormones, for example, 2,4-D, naphthalene, acetic acid, etc. Third one is postulated hormones, also produced spontaneously in plant body, but their structure, function is not discovered clearly. For example, vanillin. Classification of plant growth regulators on the basis of nature of function, how they involve in the growth of plants. E basis mele classification. First one is growth promoting hormones or growth promoters. See, these growth promoting hormones or growth promoters involves in the increase the growth of plant. So plant growth na increase madalike involve agutte. For example, auxins, gibberellins, cytokinins, etc. The second classification based on the nature of function is growth inhibiting hormones or growth retardants. See, which inhibits the growth of the plant, but they involve in other activities, other cytological, physiological, biological activity of the plant. For example, abscisic acid, ethylene, and so on. Abscisic acid involves in aging process. Ethylene is known as a fruit ripening hormone, and so on. This term auxin is derived from the Greek word auxin, means to grow or to increase. So auxin anta term Greek word inda derive agide, that means to grow or increase. Auxins may be defined as growth promoting substances which promote growth along the vertical axis when applied in low concentration to the shoots of the plant. So in a very low concentration, if you apply on the shoots of plant, it enhances the growth of the plant. See, this is a discovery regarding discovery of auxins. Of course, this discovery of auxins was successfully done by F.W. Want in 1926, and he isolated the growth substances, growth promoting substance from Havana sativa, that is oat coleoptile tips. Cole and Hagensmith in 1931 named it as auxin. Next topic is occurrence and distribution of auxins. See, these auxins occurs universally in all plant parts, irrespective of the root, shoot, leaf, and so on. See, these auxins occurs universally in all plant parts. Where there is active growth, there is auxin production. So, early meristematic activity. Active growth means meristematic activity is there. Then there should be auxin production. Growing meristems and enlarging organs produces auxins. Shoot apex produces much auxin than the root apex. So shoot region produces a comparatively larger amount of auxin when compared to root region. Apical bud synthesizes more auxin than the lateral buds. This is the comparison between the apical bud and the lateral buds. Developing seeds contain more auxin than the matured seeds. Apical bud synthesizes 6 times more auxin than the expanding leaves. So this is the occurrence and distribution of auxins in plant body. See, this auxin translocates. It moves from one part to another part by basipetally. That is auxin transported basipetally. It moves from apical to basal end. The velocity of transport, the speed, is 1 to 1.5 cm per hour in stem, and in case of coleoptiles 0.1 to 0.2 cm per hour in root region. See, these are some of the important auxins, specifically synthetic auxins produced artificially and similar to natural in their physiological activity. So auxins, some of the synthetic auxins are available in the market. These are produced artificially, but their physiological activity is similar to natural one. For example, indole propionic acid, IPA, IBA, butyric acid, naphthalene acetic acid, NAA, 2,4-dichlorophenaxy acetic acid, 2,4-D, 2,4,5-T, that is 2,4,5-trichlorophenaxy acetic acid, etc. These are all available in market. Next, effects of auxin on plant growth and development. Effect of auxin, how these auxin causes or induces plant growth and development. First thing, these auxin induces the growth of the plant by cell elongation and cell division. Definitely, cell elongation shows the growth, cell division increases the number. So both are involved, both are influenced by the auxin. The main cause of cell elongation is by increasing the osmotic content, permeability of cell to water, wall synthesis. The new material for wall will be synthesized. This is the first point. Second point, these auxins reduce the wall pressure. So internal content will be increases. Then by inducing the synthesis of RNA and protein, which in turn leads to an increase in cell wall plasticity and extension. These auxins also promotes cell division within the cambial region. See, as you all know, auxin involves in the plant growth and development. In other words, it increases the growth and development of plants. First, it increases the plant growth and development by cell elongation and cell division. And these are the different routes. These are the different routes to induce cell elongation and cell division. Next is apical dominance. Apical or terminal buds of many vascular plants are very active, while the lateral buds remain inactive. Normally, apical bud andre ಮೇಲ್ಗಡೆ ಟಾಪ್ ಅಲ್ಲಿ ಟರ್ಮಿನಲ್ ರೀಜನ್ ಇರುತ್ತಲ್ಲ, ದಟ್ ಇಸ್ ದ ಎಪಿಕಲ್ ಬಡ್. ಸೊ ಇದು ಆಕ್ಟಿವ್ ಆಗಿದ್ದಾಗ ಲ್ಯಾಟ್ರಲ್ ಬಡ್ಸ್ ಏನಿದೆ ಅದು ಗ್ರೋತ್ ಇರೋದಿಲ್ಲ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಇನ್ ಕೇಸ್ ಆಫ್ ಮಲ್ಬರಿ ಪ್ಲಾಂಟ್, ಶೂಟ್ ಟಿಪ್ ನ ಚಾಕಿಲೇರಿಂಗ್ ಆದ್ಮೇಲೆ ಶೂಟ್ ಟಿಪ್ ನ ಕ್ಲಿಪ್ ಮಾಡ್ತೀವಿ ಟು ಇಂಡ್ಯೂಸ್ ದಿ ಮೆಚುರೇಷನ್ ಆಫ್ ದಿ ರಿಮೈನಿಂಗ್ ಲೀವ್ಸ್ ಆಸ್ ವೆಲ್ ಆಸ್ ಲ್ಯಾಟ್ರಲ್ ಬಡ್ಸ್ ಗ್ರೋತ್ ಆಗಲಿ ಫಾರ್ ಸೆಕೆಂಡ್ ಕ್ರಾಪ್ ಅಂತ. ಬಿಕಾಸ್ ಈ ಎಪಿಕಲ್ ಬಡ್, ಟರ್ಮಿನಲ್ ಬಡ್ ಏನಿದೆ, ಇಟ್ ಶೋಸ್ ಎಪಿಕಲ್ ಡಾಮಿನೆನ್ಸ್ ಬೈ ಪ್ರೊಡ್ಯೂಸಿಂಗ್ ಲಾರ್ಜ್ ಕ್ವಾಂಟಿಟಿ ಆಫ್ ಆಕ್ಸಿನ್. ಲಾರ್ಜ್ ಕ್ವಾಂಟಿಟಿ ಆಕ್ಸಿನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದ್ರಿಂದ ಲ್ಯಾಟ್ರಲ್ ಬಡ್ಸ್ ಇನ್ಆಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ. ಸೊ ದಿ ರಿಮೂವಲ್ ಆಫ್ ಎಪಿಕಲ್ ಬಡ್ ಪ್ರಮೋಟ್ಸ್ ಲ್ಯಾಟ್ರಲ್ ಬಡ್ಸ್ ಟು ಗ್ರೋ. ಇದನ್ನು ನಾವು ಚಾಕಿಲೇರಿಂಗ್ ಮುಗಿತಿದ್ದ ಹಾಗೆ ಮಲ್ಬರಿ ಗಾರ್ಡನ್ ನಲ್ಲಿ ಮಾಡ್ತೀವಿ. ಎಪಿಕಲ್ ರೀಜನ್ ನ, ಟರ್ಮಿನಲ್ ರೀಜನ್ ನ ಕ್ಲಿಪ್ ಮಾಡಿದ್ರೆ ಲ್ಯಾಟ್ರಲ್ ಬಡ್ಸ್ ಗ್ರೋ ಆಗುತ್ತೆ. ಎಪಿಕಲ್ ಡಾಮಿನೆನ್ಸ್ ಈಸ್ ಡ್ಯೂ ಟು ಮಚ್ ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್, ಹೈಯರ್ ಕಂಟೆಂಟ್ ಆಫ್ ಆಕ್ಸಿನ್ when compared to lateral buds. Next point is phototropism. You know the meaning of phototropism. Phototropism is nothing but the movement of plant parts either towards or away from the source of light. On the plant parts source of light kadege ಅಥವಾ away from the source of light ಕಡೆ ಮೂವ್ ಆಗೋದನ್ನ phototropism ಅಂತಾರೆ. Shoot parts shows foliar parts. ಭೂಮಿ ಮೇಲೆ ಇರತಕ್ಕಂತ ಪ್ಲಾಂಟ್ಸ್ ಎಲ್ಲ ಪ್ಲಾಂಟ್ ಪಾರ್ಟ್ಸ್ ಪಾಸಿಟಿವ್ ಫೋಟೋಟ್ರಾಪಿಕ್ ಮೂವ್ಮೆಂಟ್ ನ ತೋರಿಸುತ್ತೆ, ಬಟ್ ರೂಟ್ಸ್ ನೆಗೆಟಿವ್ ಫೋಟೋಟ್ರಾಪಿಕ್ ಮೂವ್ಮೆಂಟ್ ಅನ್ನ ತೋರಿಸುತ್ತೆ. ಸೊ ಪ್ಲಾಂಟ್ ಬೆಂಡ್ ಟುವರ್ಡ್ಸ್ ದಿ ಯೂನಿಲ್ಯಾಟರಲ್ ಲೈಟ್ಸ್. ದಿಸ್ ಇಸ್ ಡ್ಯೂ ಟು ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಆಫ್ ಆಕ್ಸಿನ್ ಆನ್ ದಿ ಶೇಡೆಡ್ ಸೈಡ್. ಸೊ ಫೋಟೋಟ್ರೋಪಿಸಮ್ ಯಾಕಾಗುತ್ತೆ ಅಂದ್ರೆ, ಲೈಟ್ ಬೀಳೋ ಕಡೆ ಆಕ್ಸಿನ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಡಿಮೆ ಇರುತ್ತೆ, ಲೈಟ್ ಬೀಳದೆ ಇರೋ ಕಡೆ ಆಕ್ಸಿನ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಜಾಸ್ತಿ ಇರುತ್ತೆ. ಸೊ ದಟ್ ಆ ರೀಜನ್ ನಲ್ಲಿ ಸೆಲ್ ಡೆವಲಪ್ಮೆಂಟ್, ಸೆಲ್ ಇಲಾಂಗೇಷನ್, ಸೆಲ್ ಮಲ್ಟಿಪ್ಲಿಕೇಶನ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ. ಸೊ ಪ್ಲಾಂಟ್ ಮೂವ್ ಆಗುತ್ತೆ. ದಟ್ ಇಸ್ ದ ರೀಸನ್ ಫಾರ್ ಫೋಟೋಟ್ರಾಪಿಕ್ ಮೂವ್ಮೆಂಟ್ ಇನ್ ಕೇಸ್ ಆಫ್ ಪ್ಲಾಂಟ್ಸ್. ನೆಕ್ಸ್ಟ್ ಇಸ್ ಜಿಯೋಟ್ರೋಪಿಸಮ್. ಮೂವ್ಮೆಂಟ್ ಆಫ್ ಪ್ಲಾಂಟ್ ಪಾರ್ಟ್ಸ್ ಇನ್ ರೆಸ್ಪಾನ್ಸ್ ಟು ಗ್ರಾವಿಟಿ, ಗುರುತ್ವಾಕರ್ಷಣೆ, ಗ್ರಾವಿಟೇಷನ್ ಫೋರ್ಸ್ ಈಸ್ ನೋನ್ ಆಸ್ ಜಿಯೋಟ್ರೋಪಿಸಮ್ ಆರ್ ಗ್ರಾವಿಟ್ರೋಪಿಸಮ್. ಸೊ ಭೂಮಿ ಅಟ್ರಾಕ್ಷನ್ ಇದೆ, ಗ್ರಾವಿಟೇಷನ್ ಫೋರ್ಸ್ ಏನಿದೆ ಅದಕ್ಕೆ ರೆಸ್ಪಾಂಡ್ ಮಾಡೋದೇ ಜಿಯೋಟ್ರೋಪಿಸಮ್. ಪಾಸಿಟಿವ್ ಜಿಯೋಟ್ರೋಪಿಸಮ್ ಅಂದ್ರೆ ರೂಟ್ಸ್ ಶೋಸ್ ದಿ ಪಾಸಿಟಿವ್ ಜಿಯೋಟ್ರೋಪಿಕ್ ಮೂವ್ಮೆಂಟ್, ಅಂದ್ರೆ ಭೂಮಿ ಕಡೆಗೆ ಹೋಗುತ್ತೆ. ಶೂಟ್ ಪಾರ್ಟ್ಸ್ ಏನಿದೆ ದಟ್ ಶೋಸ್ ನೆಗೆಟಿವ್ ಜಿಯೋಟ್ರೋಪಿಕ್ ಮೂವ್ಮೆಂಟ್. ಸಿ, ಸ್ಟೆಮ್ ಅಂಡ್ ರೂಟ್ಸ್ accumulate indole acetic acid in the lower side of in the response to gravity. So increasing the concentration of the lower side of the stems causes those cells to grow more than the cells on the upper side. See, ಒಂದು ಪ್ಲಾಂಟ್ ಅನ್ನ ಲ್ಯಾಟರಲ್ ಆಗಿ ಮಲಗಿಸ್ಬಿಟ್ರೆ, ಒಂದು ಪ್ಲಾಂಟ್ ಅನ್ನ ಹೀಗೆ ಕರ್ಸರ್ ಹೀಗೆ ಒಂದು ಪ್ಲಾಂಟ್ ಅನ್ನ ಮಲಗಿಸ್ಬಿಟ್ರೆ, ಸೊ ಲೈಟ್ ಬೀಳ್ತಾ ಇದ್ದಾಗ ಏನಾಗುತ್ತೆ, ಈ ಶೂಟ್ಸ್ ಮೇಲ್ಗಡೆ ಟರ್ನ್ ಆಗುತ್ತೆ, ರೂಟ್ಸ್ ಕೆಳಗಡೆ ಟರ್ನ್ ಆಗುತ್ತೆ. ಬಿಕಾಸ್ ಕೆಳಗಡೆ ಇಟ್ಟಾಗ ಮೇಲ್ಗಡೆಯಿಂದ ಲೈಟ್ ಬೀಳ್ತಾ ಇರುತ್ತೆ, ಲೈಟ್ ಬೀಳ್ತಿದ್ದಾಗ ಲೈಟ್ ಇರೋ ಕಡೆ ಆಕ್ಸಿಜನ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಡಿಮೆ ಇರುತ್ತೆ. ಸೊ ಎಲ್ಲ ಬಾಟಮ್ ಅಲ್ಲಿ ಸೆಟ್ಲ್ ಆಗಿರುತ್ತೆ. ಶೂಟ್ ರೀಜನ್ ಅಲ್ಲಿ ಸೆಲ್ ಡಿವಿಷನ್ ಗ್ರೋತ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ. ಸೊ ಇಟ್ ಬೆಂಡ್ ಟುವರ್ಡ್ಸ್ ದಿ ಅಪ್ಪರ್ ಸೈಡ್. ಬಟ್ ಲೋವರ್ ಪೋರ್ಷನ್ ಅಥವಾ ರೂಟ್ ರೀಜನ್ ಅಲ್ಲಿ ಅದು ಸೆಲ್ ಡಿವಿಷನ್ ಕಡಿಮೆ ಮಾಡುತ್ತೆ. ಸಿ, ದಿಸ್ ಇಸ್ ದ ಆಕ್ಷನ್ ಆಫ್ ಆಕ್ಸಿನ್. ಎರಡು ರೀತಿ ವರ್ಕ್ ಮಾಡುತ್ತೆ. ಶೂಟ್ ರೀಜನ್ ಅಲ್ಲಿ ಸೆಲ್ ಡಿವಿಷನ್ ನ ಹೆಚ್ಚು ಮಾಡುತ್ತೆ, ರೂಟ್ ರೀಜನ್ ಅಲ್ಲಿ ಸೆಲ್ ಡಿವಿಷನ್ ಕಡಿಮೆ ಮಾಡುತ್ತೆ. ದಟ್ ಈಸ್ ದ ರೀಸನ್, ದಟ್ ಈಸ್ ದ ಸೈಂಟಿಫಿಕ್ ರೀಸನ್ ಫಾರ್ ಜಿಯೋಟ್ರೋಪಿಕ್ ಮೂವ್ಮೆಂಟ್. ಪಾಸಿಟಿವ್ ಜಿಯೋಟ್ರೋಪಿಸಮ್ ಇರ್ಬೋದು ಅಥವಾ ನೆಗೆಟಿವ್ ಜಿಯೋಟ್ರೋಪಿಸಮ್ ಮೂವ್ಮೆಂಟ್ ಇರ್ಬೋದು. ಸೊ ದಟ್ ಈಸ್ ಡ್ಯೂ ಟು ದಿ different concentration of indole acetic acid. Effect of auxin on plant growth and development continuing. Root initiation. Of course, application of indole acetic acid to cut end of stem promotes the root formation. ಇದನ್ನ ನಾವು ಮಲ್ಬರಿನಲ್ಲಿ ರೂಟಿಂಗ್ ಪ್ರಾಬ್ಲಮ್ ಇರತಕ್ಕಂಥ ಮಲ್ಬರಿ ಕಟಿಂಗ್ಸ್ಗೆ ಇದನ್ನು ಅಪ್ಲೈ ಮಾಡ್ತೀವಿ. ವಿಲ್ ಆಲ್ಸೋ conduct this experiment in your practical class. Control or prevention of abscission. Abscission ಅಂದ್ರೆ ಏಜಿಂಗ್. ಸೊ ಆಕ್ಸಿನ್ ನೀವು ಅಪ್ಲೈ ಮಾಡ್ತಾ ಇದ್ರೆ ಸೊ ಇದು ವೆಜಿಟೇಟಿವ್ ಗ್ರೋತ್ ನ ಯಂಗ್ನೆಸ್ ನ ಕೊಡ್ತಾ ಇರುತ್ತೆ, ಗ್ರೋತ್ ಆಗ್ತಾ ಇರುತ್ತೆ ಆಕ್ಸಿನ್. ಸೊ ಹಾಗಾಗಿ ಅಬ್ಸಿಷನ್ ಡಸ್ ನಾಟ್ ಅಕರ್ ವೆನ್ ಆಕ್ಸಿಜನ್ ಕಂಟೆಂಟ್ ಈಸ್ ಹೈ ಆನ್ ಡಿಸ್ಟಲ್ ಎಂಡ್, ಲೋ ಇನ್ ದ ಪ್ರಾಕ್ಸಿಮಲ್ ಎಂಡ್ ಆಫ್ ದಿ ಅಬ್ಸಿಷನ್ ಝೋನ್. ಎಲ್ಲಿ ಏಜಿಂಗ್ ಪ್ರಾಸೆಸಿಂಗ್ ಇದೆ ಅಲ್ಲಿಗೆ ಈ ಆಕ್ಸಿನ್ ಎಕ್ಸೋಜಿನಸ್ ಅಪ್ಲಿಕೇಶನ್ ಆಗ್ತಾ ಇದ್ರೆ ಇಟ್ ಪೋಸ್ಟ್ಪೋನ್ಸ್ ದಿ ಅಬ್ಸಿಷನ್ ಪ್ರಾಸೆಸ್. ನೆಕ್ಸ್ಟ್, ಕ್ಯಾಲಸ್ ಫಾರ್ಮೇಶನ್. ಕ್ಯಾಲಸ್ ಇಸ್ ನಥಿಂಗ್ ಬಟ್ ದಿ a mass of undifferentiated parenchymatous tissue or cells. ಸೊ ಈ ಕ್ಯಾಲಸ್ ಫಾರ್ಮೇಶನ್ ಇಂಡ್ಯೂಸ್ ಮಾಡುತ್ತೆ. ಹಾಗಾಗಿ ಪ್ಲಾಂಟ್ ಟಿಶ್ಯೂ ಕಲ್ಚರ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ಸ್ ಅಲ್ಲಿ ಇದನ್ನ ಯೂಸ್ ಮಾಡುತ್ತೆ. ಸೆಕ್ಸ್ ಎಕ್ಸ್ಪ್ರೆಷನ್. ಆಕ್ಸಿನ್ ಇಂಡ್ಯೂಸ್ಡ್ ದಿ ಚೇಂಜಿಂಗ್ ಆಫ್ ಸೆಕ್ಸ್ ರೇಷಿಯೋ ಇನ್ ಫ್ಲವರ್ಸ್ ಟುವರ್ಡ್ಸ್ ದಿ ಫಿಮೇಲ್ನೆಸ್. ದಟ್ ಈಸ್ ಇಟ್ ಇನ್ಕ್ರೀಸಸ್ ದಿ ನಂಬರ್ ಆಫ್ ಫಿಮೇಲ್ ಫ್ಲವರ್ಸ್. ಸೊ ಆಕ್ಸಿನ್ ಜಾಸ್ತಿ ಇದೆ ಅಂತ ಅಂದ್ರೆ, ಆಕ್ಸಿನ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಇದೆ ಅಂದ್ರೆ ಇಟ್ ಹೆಲ್ಪ್ಸ್, ಇಟ್ ಇನ್ಕ್ರೀಸಸ್ ದಿ ನಂಬರ್ ಆಫ್ ಫಿಮೇಲ್ ಫ್ಲವರ್ಸ್, ಫಿಮೇಲ್ನೆಸ್ ಅನ್ನ ಜಾಸ್ತಿ ಮಾಡುತ್ತೆ. ಅಪ್ಲಿಕೇಶನ್ ಆಫ್ naphthalene acidic acid in case of mango. Indole butyric acid in case of guava in stem cuttings causes 100% success in vegetative propagation. So rooting problem iddaga auxins. Naphthalene acidic acid is also a type of auxin. Indole butyric acid is also a type of auxin. So these are specific. Mango nalli NAA, guava dalli IBA apply ಮಾಡಿದ್ರೆ 100% success in vegetative propagation. It prevents sprouting. Seedless fruit production, that is nothing but the parthenocarpy. Parthenocarpy is nothing but the pollination and fertilization without pollen grains. So ಮೇಲ್ ರೋಲ್ ಏನು ಇಲ್ದೇನೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಆಗೋದನ್ನ ಪಾರ್ಥೆನೋಕಾರ್ಪಿ ಅಂತ ಕರೀತಾರೆ. ಸೊ ಸೀಡ್ಲೆಸ್ ಫ್ರೂಟ್ ಪ್ರೊಡಕ್ಷನ್ ಅಲ್ಲಿ ದಿಸ್ ವಿಲ್ ಬಿ ಯೂಸ್ಡ್. ಇನ್ ಕೇಸ್ ಆಫ್ ಬನಾನಾ, grapes, strawberry, brinjal, grapes, application of indole acetic acid, IBA and NAA shows 100% success. That is for the production of seedless fruits. In case of agriculture, for promotion of flowers, application of NAA causes uniform flowering in pineapple, leading to development of uniform sized fruits. So iದೇನಾಗುತ್ತೆ ಅಂದ್ರೆ ಫ್ಲವರಿಂಗ್ ಅನ್ನ ಪ್ರಮೋಟ್ ಮಾಡುತ್ತೆ, ಯೂನಿಫಾರ್ಮಿಟಿ ಇರುತ್ತೆ at a time. Because crop harvesting, it will be very useful, helpful. 2,4-D is also used to increase the femaleness in monoecious cucurbits. Then it prevents of premature dropping of fruits. Premature dropping. In case of apple and cotton, naphthalene acidic acid is generally used. In case of citrus fruits, 2,4-D, 2,4,5-T are generally used. For germination purpose, indole acetic acid, indole butyric acid is most widely used in soaking seeds for germination. In case of fruit setting, 2,4,5-T is used to improve fruit setting in berries. Thinning of flowers, fruits and leaves, 2,4-D is used for defoliation of cotton plant before harvesting. Because ಕಾಟನ್ ಹಾರ್ವೆಸ್ಟ್ ಮಾಡಬೇಕು ಅಂದ್ರೆ ಲೀಫ್ ಇರಬಾರ್ದು. ಸೊ ಆಗೇನ್ ಮಾಡ್ತಾರೆ, 2,4-D ಅಪ್ಲೈ ಮಾಡಿದಾಗ ಡಿಫೋಲಿಯೇಷನ್ ಆಗುತ್ತೆ, ಇರೋ ಎಲೆ ಎಲ್ಲ ಉದುರೋಗುತ್ತೆ, ಕಾಟನ್ನ ಈಸಿಯಾಗಿ ಬೈ ಮೆಷಿನ್ ದಟ್ ವಿಲ್ ಬಿ ಹಾರ್ವೆಸ್ಟೆಡ್. Naphthalene acidic acid is used for fruit thinning in apples. Because ಹೆಚ್ಚಿಗೆ ನಂಬರ್ ಇದ್ರೆ ಗುಡ್ ಕ್ವಾಲಿಟಿ ಬರೋದಿಲ್ಲ, ಹಾಗಾಗಿ ಒಂದಷ್ಟು ಥಿನ್ನಿಂಗ್ ಇಸ್ ವೆರಿ ಎಸೆನ್ಷಿಯಲ್. ಸೊ ನಾರ್ಮಲಿ ದೀಸ್ ಪ್ಲಾಂಟ್ ಹಾರ್ಮೋನ್ಸ್ ಆರ್ ಯೂಸ್ಡ್. To remove the weeds, of course there are several methods. but using plant hormones, 2,4-D, MC, PA, methyl chlorophenoxyactic acid are weed killers. So weed ಜಾಗದಲ್ಲಿ ಅಪ್ಲೈ ಮಾಡಿದ್ರೆ ಡೆತ್ ಆಗುತ್ತೆ. 2,4-D is highly toxic to broad-leaved plants or dicotyledons. So monoculture ಕಲ್ಚರ್ನಲ್ಲಿ 2,4-D ನ ಯೂಸ್ ಮಾಡಿದ್ರೆ dicotyledons weeds ಏನಿದೆ that will be removed. In case of tissue culture, auxin along with cytokinin shows successful callus formation, root, shoot differentiation, and so on. The second one is gibberellins. See, auxin is first type of phytohormone, plant hormone. Gibberellins is another class of phytohormone. This gibberellins discovered by Kurosawa, a Japanese plant pathologist, in 1928. Rice plants infected by the fungus Gibberella fusarium, that is Fusarium moniliforme, showed excessive stem elongation, and this symptom is called Beckonet disease, foolish seedling anta ಕರೆದ beginning ಅಲ್ಲಿ. But later ಅದು ಗೊತ್ತಾಯಿತು because of the fungus association ಅಂತ. Then the chemical was extracted and purified and named as gibberellic acid. Now more than 80 different gibberellins, different types of gibberellins are available in the market. They are named as GA1, gibberellic acid 1, 2, 3, and so on. The most commonly occurring gibberellins is gibberellic acid 3. See, these are some of the important physiological effects of gibberellins. Number 1, removes genetic dwarfism. Due to genetic makeup, some plants show dwarfism. But if you apply the exogenous application of gibberellins, removes the genetic dwarfism, so the dwarf plants may grow normally. Then bolting and flowering, it improves bolting and flowering. Light inhibited stem growth, it removes the light inhibited stem growth. Induces parthenocarpy. Mobilization of storage compounds during germination. During germination, due to incompatibility between the embryo and endosperm, germination may be failed. During such conditions, exogenous application of gibberellins leads to good germination percentage. Next class of plant hormone is cytokinins. Heberland in 1913 noticed a soluble substance in the pith of potato tubers which possesses the capacity to induce cell division. Cell division induce ಮಾಡ್ತಕ್ಕಂಥ ಎಬಿಲಿಟಿ ಇರೋ ಒಂದು ಸಬ್ಸ್ಟೆನ್ಸ್ ಅನ್ನ ಹೇಬರ್ಲ್ಯಾಂಡ್ ಐಡೆಂಟಿಫೈ ಮಾಡ್ತಾರೆ. In 1939, Bonner isolated the traumatic acid from bean fruits which can induce the meristematic activity when a tissue is injured. Skoog in 1948 worked on the stem internode culture of tobacco plant. Miller et al. In 1955 isolated another substance from yeast and named as kinetin. It was effective in induction of cell division in very low concentration. Of course, hormone antidagane, the concentration should be very, very low or less. Subsequently to its discovery, many analogues of kinetin active in promoting cell division were synthesized. These cytokinins involves in cell division, cell enlargement, root initiation and growth, shoot initiation and growth, breaking of dormancy, and so on. Kinetin is a degraded product of DNA. So deoxyribonucleic acid degrade ಆದಮೇಲೆ kinetin produce ಆಗುತ್ತೆ. That is a type of kinetin. Next, till now, auxin, gibberellin, cytokinins are the growth promoters. Growth promoters. But some growth retardants, inhibitors are also there. First one is ethylene. Ethylene is a growth retardant. It prevents the growth, but it promotes ripening, fruit ripening process. So growth retard ಆಯಿತು ಅಂದ್ರೆ ಪ್ಲಾಂಟ್ಗೆ ನೆಗೆಟಿವ್ ಇಂಪ್ಯಾಕ್ಟ್ ಅಂತ ಅಲ್ಲ. ಸೊ ಇದು ಮೆಚುರೇಷನ್ಗೆ, ಫ್ರೂಟ್ ರೈಪನಿಂಗ್ಗೆ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಹಾಗೆ ಅಬ್ಸಿಸಿಕ್ ಆಸಿಡ್, ಅನದರ್ ಗ್ರೋತ್ ರಿಟಾರ್ಡೆಂಟ್, ಇಂಡ್ಯೂಸಸ್ ಸ್ಟೊಮ್ಯಾಟಾ ಕ್ಲೋಸಿಂಗ್. ಸ್ಟೊಮ್ಯಾಟಾ ಕ್ಲೋಸ್ ಮಾಡುತ್ತೆ. ಸೊ ಇಟ್ ಇಂಡ್ಯೂಸಸ್ ದಿ growth retardation, but it inhibits bud growth and shoot formation. Bud growth inhibit ಮಾಡುತ್ತೆ, shoot formation ಮಾಡುತ್ತೆ, but it controls the aging process. ಒಂದು ಫ್ರೂಟ್ ರೈಪ್ ಆಗ್ತಾ ಇದೆ, ರೈಪ್ ಆಗಿದೆ, ಅಬ್ಸಿಷನ್ ಆಗಬೇಕು, ಫಾಲ್ ಆಗಬೇಕು ಅಂದಾಗ ಇದು ಆಕ್ಟ್ ಮಾಡುತ್ತೆ. ಸೊ ಗ್ರೋತ್ ರಿಟಾರ್ಡೆಂಟ್ಸ್ ಆರ್ not don't have any negative effect on plants, but they have some other than the growth, they have some physiological role like maturation, fruit ripening, and so on. Thank you.